Okay, so 2.3 carbohydrates and lipids. I'll have this one concept map for carbohydrates and then another one page for lipids. So let's begin with carbohydrates. Just a quick review, it comes from the word carbo and hydrates. Hydrates meaning water, and carbo from carbon. So in a ratio for a carbohydrate, there's 1 carbon for every 2 hydrogen for every 1 oxygen. So what we need to learn here are the different types and structure of carbohydrates. So let's begin by talking about the types of carbohydrates. We can divide this into 3 different types. Of your mono, di, and polysaccharides. Mono comes from the word one, di from the word two, and poly from the word many. Let's begin. We have 3 kinds of monosaccharides, or 1 sugar unit type of carbohydrates. We have glucose, fructose, and galactose. Now, if we combine glucose with fructose, with the process of condensation, we get a disaccharide called sucrose. That's our table sugar, and this also happens to have the highest sweetness level amongst the different types of sugars here. Now, if I combine glucose to galactose, we can form a disaccharide called lactose. So this is the carbohydrate found in your milk. And if we have glucose combined to itself, we'll just put an arrow like that, we form a disaccharide called maltose. A quick way to remember this is to remember the mnemonic, my gorgeous girlfriend is slim. Is slim. So, gorgeous girlfriend are your monosaccharides, slim are your disaccharides, sucrose, lactose, and maltose. All you have to remember is to memorize which monosaccharides combine to which monosaccharide to form these disaccharides. Okay? So we have 3 monosaccharides that we need to know, 3 disaccharides formed from those monosaccharides, and also 3 polysaccharides. So we have cellulose, we have starch, and we have glycogen. So before I discuss the different structures of cellulose, starch, and glycogen, let me just have a quick review about the structure of glucose. So this is a ring of glucose, and you already learned that we start numbering from carbon 1 here, carbon 2, carbon 3, carbon 4, carbon 5, and H2OH right there contains your carbon 6. Now, 3 carbons that you need to know here are the positions of carbon 1, carbon 4, and carbon 6. So, for cellulose, this is formed by combining many glucose units, and a beta 1,4 bond is formed between those glucose units. So how would that look like? A beta means that the hydroxyl found in carbon 1 is here on top. So that's a beta glucose, and alpha glucose is where the OH is found here, downward, so upward and downward. So for glucose 1,4, a beta glucose, so it would look something like this. OH is here on top, and it will combine with carbon 4 of the next glucose molecule, carbon 1, 2, 3, 4 here. So if I can actually move this up, they should be at least 180 degrees next to each other, and we remove that and release water in the process of condensation, and you already know that the bond formed between these two glucose molecules is called a glycosidic bond. So for starch, what you need to know here are, it's made up of, again, many glucose subunits, but this time they are connected. The connections are found between carbon 1 of the first glucose to carbon 4 of the next glucose, but they are forming an alpha bond. And I've mentioned earlier, an alpha glucose is when your hydroxyl in carbon 1 is found here in the bottom. So let's draw that again. This is carbon 1, and the OH is right here. And it will bond with another glucose. 1, 2, 3, 4. The OH is also right here. And water is released, and another bond is formed. So an alpha 1,4 bond and a beta 1,4 bond. So because of the how your glucose molecules are oriented, in a beta 1,4 bond, they're 180 degrees of each other, and that will eventually form a straight chain of a polysaccharide. And for your starch, since they are both oriented downwards, so they will eventually, as you keep adding more glucose, form sort of like a curved polysaccharide. And for starch, you also have to know that there's actually two types of starch molecules. So sorry, there's space here. So you have your amylose and amylopectin. Both contains alpha 1,4, but amylopectin has an additional bond of alpha 1,6. You can just easily guess this. It's basically a bond between carbon 1 of the first glucose and carbon 6 of the next glucose. This results into branching of your polysaccharide molecule. So you have this curved, or it's sort of like a spiral molecule, and then on the sides it will start branching because of the alpha 1,6. And this amylopectin is the same structure as your glycogen. So glycogen has alpha 1,4 and also alpha 1,6, but with many more branching, many more alpha 1,6. Therefore, the overall structure of your glycogen is actually more compact compared to this amylopectin. So here in this picture, you can see the difference between beta and alpha glucose molecules. So for cellulose, again, made up of beta glucose molecules, the OH is oriented upward. So this is how they form, and this is like right side up, and the next glucose is upside down. That's what I meant when I said they're 180 degrees next to each other. This forms a straight chain. Then you have here starch. As an example, here is an alpha glucose, so the OH is here downward, and since it's also downward on carbon 4 of the next glucose, they're just aligned, and this will eventually form a ring. The same alpha 1,4 bond is found in glycogen. Now let me show you an alpha 1,6 in your amylopectin and also in your glycogen. Okay, so you have here, let's look at the picture here on the top. We have your alpha 1,4 bonds, and then the 6th carbon of this glucose molecule, carbon 1, 2, 3, 4, 5, 6. So carbon 6 bonds with carbon 1 of another glucose molecule here. So this alpha 1,6 is actually responsible of forming the side branches of this macromolecule. Another picture here in the bottom, you see your alpha 1,4, and then here you have your branching caused by an alpha 1,6 bond. So these alpha 1,6 is only present in this type of starch, which is amylopectin, and also in glycogen. There's more alpha-1,6 in your glycogen, hence it's more compact in its structure compared to amylopectin. So let us end with the different functions of your 3 polysaccharides here. You already know the structure. Now let's talk about the function. Now for your cellulose, as I've said earlier, they orient themselves upside down next to each other. They're able to form straight chains of your polysaccharide molecule. And these straight chains can eventually form bundles, and we can call them cellulose microfibrils. And we find these molecules, we find these cellulose molecules as part of the cell wall of your plant cells, and it provides your plant cell tensile strength. So basically, if you remember how your plant cells look like under the microscope, they're very regularly shaped, and the plant cells normally keep their shape, and they try to withstand extreme pressure, osmotic pressure changes inside the plant. So that's the purpose of your cellulose. And just a side note, humans cannot produce the enzyme called cellulase, and we therefore cannot digest cellulose properly. So if our food is rich in vegetables and fruits, which are all plants and they contain plant cells, we might not be able to digest them, and you already know sometimes your food might contain portions of your corn, or I don't know, any other vegetable that you've eaten. So this is a very strong type of macromolecule. We are not able to digest this. This gives shape and structure to plant cells. And your starch is also related to your plants. So starch is the form of macromolecule where your plants store their energy. So as plants produce glucose in the process of photosynthesis, they have to keep it in a storage form of starch, because glucose is easily soluble in water, but starch is not. It's a bigger molecule, so they store this in their sinks, such as the roots or actively growing parts of the plant. And then your glycogen, so for the function of your glycogen, this is made by us, humans and animals, and we actually, if there's excess glucose in our diet, we take it away from the blood circulation, and we link those glucose molecules and store it as glycogen in the liver. Vice versa, if you need glucose, let's say in moments of fasting, times of fasting, you need glucose, so you break it apart, you remove the bond, and you make available that glucose into your blood. So cellulose and starch, they serve their function for the plants, and glycogen, it serves its function for humans or animals. That's our form of storage of this carbohydrate.